Protein making in progress. So one of the most common ways to get a lot of a protein that we want to study is to stick the instructions for making the protein into bacteria and ask the bacteria to make it for us. The really cool thing about doing this in bacteria is that we can use this like inducible expression system where we get the bacteria to make a lot of it on demand. And so we call this like recombinant protein overexpression using this like T7 system. And so I'll tell you more about it, but the basic idea is that we stick the genetic instructions into a circular piece of DNA called a plasmid, and this is we put it in a place where it's under control of this T7 promoter. So T7 is a polymer, an RNA polymerase, and so it's going to make RNA copies of your gene when the T7 polymerase is there. And then we control the expression of the T7 polymerase by putting this plasmid into special cells where that T7 polymerase is under the control of the lac operon, which is, you can trick it into getting activated when you add a chemical called IPTG. So when you add IPTG, now that starts making the T7 polymerase. The T7 polymerase goes to that promoter in front of your gene, starts making mRNA. Now in bacteria, transcription, so the making of the mRNA, and translation, the making of the protein, are coupled. So once it starts making the mRNA, now it's going to start making the protein. And voila, you've got a lot of protein. And I talk about a lot because like T7, it's like this phage, it's this bacteria infecting virus. And so it's really good at making a lot of things. And so if you put this T7 promoter in front of your thing, and then you add your gene, and then you have the T7 promoter polymerase around, the bacteria is gonna like basically stop doing everything else and start making a ton of your protein. So it like overexpresses it. So it's really great if you have like a simple protein that bacteria don't really have a problem with folding or modifying or any of that stuff that you need to worry about for more complicated proteins, in which cases you can use things like insect cell expression that I talked about yesterday. But for other things, bacteria is cheap, easy, much quicker, and it can be a really great option, and you see, so it's one of the most common ways that proteins are expressed like recombinantly. So let's go into more detail about basically the background as well as the practical aspects and how you actually go about doing this. So the instructions for making proteins are written in the form of genes, which are DNA. And then the machinery that makes the protein are ribosomes, and what they read, they work with messenger RNA, which are RNA copies of the genes. So the gene gets transcribed into this messenger RNA, and then this messenger RNA gets translated into protein. So in this process of translation, the ribosome is going to move along the RNA, and it's going to put in the corresponding amino acids, so protein letters. And this genetic code is universal. So basically, no matter where the genetic instructions come from, the ribosome is able to interpret them and make a protein. So even if we stick a human gene into bacteria, we can still get bacteria to make it for us. So the idea with recombinant protein expression, I'll get more into this idea of what recombinant means in a second, but you can stick the genetic instructions for making the protein into a circular piece of DNA called a plasmid, and you stick that into bacteria, and the RNA polymerases in the bacteria will start making mRNA copies, and then the ribosomes of the bacteria will start making protein from those copies. And so you have your protein made in these bacteria. And what's really great about bacterial systems, as we'll get into, is that we can use this inducible system where we can get them to make the protein we want on demand. So it's really, really cool. So they don't make the protein, and then they're all growing and happy, and then we induce them. So we add this chemical, and then they stop growing, but we've let them grow so we have enough of them, and then they start making oodles of our protein, which is really, really great. So let's get into a little of the terminology before we try to go too deep into what's going on. So I keep saying like genetic instructions as opposed to like gene, because what we're putting in is actually we're putting in the complementary DNA or the cDNA. The messenger RNA is a copy of the gene, but it's not
letter to letter. I mean, the parts that are left in are letter to letter, but a lot of parts are cut out. So there's these regulatory regions called introns. Those introns are really important for regulating transcription and that sort of thing. But once you get to the messenger RNA stage, the ribosome isn't going to know what's an intron, so that regulatory information, and what's an exon, so that information that actually has instructions for making proteins. And so those introns are going to get cut out in a process called splicing. And this isn't going to happen in your bacteria. And even if it did, it might happen kind of differently because there's this thing called like alternative splicing. So we're not going to get into that. The basic thing is that we want the mature messenger RNA version. So in the mature messenger RNA, those introns have been cut out. But we want the DNA version of this mature messenger RNA, and we call this the complementary DNA. And so the cDNA is what we're actually putting in so that we get the final version of the protein that we want. And we can also make modifications to this. We can make mutations and that sort of thing if we want to make differences in the protein to see what various parts of the protein do. So really cool stuff. So now getting back to that recombinant term. So recombinant, basically we're just recombining bits of genetic information. And so we're recombining the DNA, the cDNA, with the DNA of this plasmid vector. And so we just call a vector something that we can use as like a vehicle. And a plasmid is one of these like circular pieces of DNA that can be hosted inside of a bacterial cell outside of the bacteria's own genetic information. So the bacteria has its own like genomic DNA, that's like its own chromosomes, that sort of thing. And then it can hold this like plasmids on the side. And we call this recombinant because we're recombining. And we can do this with different molecular cloning techniques. So we can I use like PCR-based methods like SLIC. You can also use your restriction enzymes. So you make cut out your gene or your cDNA, and then you cut out the region in the plasmid, and then you stitch them together with DNA ligase. So there's different ways that you can do this cloning part. But you want to stick it into a plasmid that is going to allow it to be expressed, and it's going to allow it, if that's what your goal is at this point, or it's going to allow it to at least be replicated within the bacteria. So this is where this origin of replication, this ORI, comes in. So this is where so that the plasmid is going to get replicated, and it's not just going to, so it'll get passed on. So every time the bacteria makes a copy of itself, it can also make a copy of this plasmid, so the plasmid gets passed on. And you can actually have like high copy numbers, so you can have more than one copy of the plasmid in the cells, which is good for the cloning phase when you're just trying to get a lot of the plasmid. So basically, the vector plasmid, in addition to having that origin, you're going to have an antibiotic selection, so some sort of selection marker, typically an antibiotic resistance gene. And this is going to allow us to then grow the cells in media that has the antibiotic in it. And this way, if only the cells that have the plasmid, that have the antibiotic, will be able to grow. And so this is going to allow us to select for them. And we also, when it comes to the actual expression part, so those things we need no matter what we're doing with the plasmid. But if we want the plasmid to be used to make a protein, we need to have some sort of promoter in front of our gene. I'm just gonna say gene, but it's, with the caveat that it's not the actual gene, it's like this edited version of the gene. But it's a lot easier to kind of conceptualize and just say, and I tend to just like slip into saying it, and we tend to say it. It's just a caveat to keep in mind if you're doing this cloning, that's not the actual gene as it was in its original home. So, but it does need a promoter, and this promoter is usually different than the promoter that was in its original home. And so the promoter is the sequence that's going to tell the RNA polymerase to start making messenger RNA copies of your gene. And so you need this promoter to tell them to make these copies. So in this T7 system, the promoter that we use is for the T7 polymerase. So T7 is a phage. It's a bacteria. Bacteriophage is the virus that infects bacteria. And it infects bacteria and it gets bacteria to start making a lot of it. And in order to do this, it actually uses its own promoter. So it uses its own promoter and it uses its own RNA polymerase. This way, it doesn't have to worry about competing with all of the bacterial mRNAs, which use these bacterial promoters and the bacterial RNA polymerase. So it gets, as long as it can get the bacteria to make this RNA polymerase and it has all its genes under control of its own RNA polymerase, now even if the bacteria is all RNA polymerase are all sidetracked with all the bacterial RNAs, or the bacteria is like not doing so well because it has all this virus, this is still going strong. And so it's able to, it has this really strong promoter that's able to induce a high level of expression. And so if we stick this promoter in front of our gene, and if we put them in cells that make this polymerase, then we can get a high yield of our protein. We can also do this just to get the RNA. So this is how like in vitro transcription works. But this post, we're actually doing this inside of bacteria. And so we're going to have the bacteria use this RNA to make a protein. So we stick this T7 RNA polymerase promoter in front of our gene. And often this is done with a PET vector. So a plasmid, PET stands for plasmid expression vector under T7 control, because it's under the control of the T7 polymerase. So as long as we put a T7 promoter in front, and then we also have a T7 terminator in the end. So the T7 promoter is going to tell the T7 RNA polymerase to latch on and start transcribing the DNA into RNA. And then the terminator, it folds up into this like hairpiny thing and says like, stop, you've reached the end of the gene. And what's really cool is that in bacteria, transcription and translation are coupled. So it starts making this protein as well as still making the mRNA. Really cool. In our cells, the transcription and translation are separated. So we have the transcription occurring in the nucleus, so in a membrane-bound compartment in the cell. And then the messenger RNA, the processing to make the mature messenger RNA, that all happens in the nucleus, and then that gets shipped out into the cytoplasm, so the general cellular interior, which is where translation is going to occur. But in bacteria, these are prokaryotic. They don't have like this nucleus with the separation of everything. You have this big room in the bacteria where this can all happen. So the translation can happen co-transcriptionally. So as long as we can control that, as long as we can control the production of the transcription, then we can control the translation. And how we're going to control the transcription is going to be by controlling when we're going to express the RNA polymerase. So that's what's coming up. But first, we need to get this actually into those cells. So we need to get it into the cells that have the instructions for making the T7 polymerase. Because if you just have cells that don't have, if cells don't have this T7 polymerase, because I mean, like E. coli wouldn't normally make that, because if it's not infected by this virus. So we need to use special strains of E. coli that actually have this. And so like BL21 DE3 cells. So the DE3 cassette has the T7 RNA polymerase under the lac operon control. This is going to be the inducible control system. So it's going to only make it when you add the IPTG. So basically how this works is that this lac operon is this thing in bacteria that they use to control the expression of the beta-galactosidase, so beta-gal. And so this is this enzyme or this reaction speeder upper. And what it does is it breaks lactose into glucose and galactose. And bacteria only make it when they need to make it. So they prefer to use glucose and if they don't have to break down lactose to get the glucose they want if they can just get glucose made by itself. So if like there's plenty of glucose around, they're not going to want to make this because it's going to take energy to break this down to get glucose when they could just use the glucose that's already there. It also takes energy to make this beta-galactosidase, and so you don't want to use your resources to make this if you don't need to, and you also don't need to. So you don't need to make it if you have glucose around, and you also don't need to make it if you don't have lactose around. And so it's under this system of control where normally it's not a problem. You don't need to have this made because you've got plenty of glucose, not a lot of lactose. And so there's this lac repressor that's sitting on top of this lac promoter. And so that is in front of the gene, this LAC-Z gene for making beta-galactosidase. So this is like the lac promoter. And normally this is protein called the lac repressor. And so the lac repressor is going to be sitting on top of here and preventing RNA polymerase from coming and binding and from making the messenger RNA, which then gets made into the beta-galactosidase protein. So, but you need to be able to, so normally it is repressed, so nothing's being expressed. But when there is a lot of lactose, you want to make this. And so what happens is how this happens is that the repressor is going to fall off. And it's not just going to fall off because it suddenly loses its interest. It's going to lose interest because it binds to allolactose and kind of undergoes a conformational change, so like a shapeshift. So this allolactose molecule, it's made from lactose. When there's a lot of lactose, some of it gets rearranged into this allolactose, and then this allolactose can bind to the lac repressor. So this protein that's blocking the site, and then the repressor falls off, and so now you can get this expressed. So you can imagine that if we stick something else here instead of this LAC-Z gene, we stick this promoter in front of it, we can still use this repressive system, this inducible derepression to get the thing that we want made. So instead of using allolactose or lactose, we use IPTG, which is this molecule that mimics allolactose. And so it will cause this to derepress, but it's not subject to like breakdown and stuff like the lactose or the allolactose were. So we don't have to keep adding it. We just have to like add it once. What's going to happen is that when we stick in the bacterial strains that have this T7. So we're talking about the induction is going to be of this T7. So we've replaced the beta-galactosidase gene with the gene for T7 RNA polymerase. And this is happening in the host bacterial DNA. So this is not happening in our plasmid that we put in. Our plasmid, though, is relying on the T7 RNA polymerase made from this inducible system. So we're controlling the expression of T7 RNA polymerase, and thereby we can control the expression of our gene. So our gene is under the T7 RNA polymerase promoter, and the RNA polymerase is under the LAC promoter. So what's going to happen is that when we induce the system, so normally the LAC repressor is sitting on top of here. Then when we add IPTG, this is like we were adding allolactose to the bacteria. They don't know the difference. That's going to bind, it's going to de-repress this. So T7 polymerase is going to get made. Then that T7 polymerase is going to be used in our vector to make this protein. And so the gene is going to get transcribed and the mRNA is going to get translated and we get our protein, and it's super duper awesome. And because this T7 promoter is so strong, we're going to get a lot of this. Because this T7 promoter is so strong, the one that we put in front of our gene, we're going to get a lot of this made. So typically we can get the bacteria to make like 50% of their protein that they make is going to be our protein. If it works out well, we get a good overexpression. It's really, really awesome. So this is why we want to have it under this inducible system though as well, because we don't want to have the cells make it before there's enough cells, because you can imagine this is going to be really resource intensive for these bacteria, and they're not going to be able to be growing when they're spending all of their energy making this protein. They're not going to just be like splitting and reproducing and stuff. So we want to induce them once we have a high enough quantity of the bacterial cells, and so we monitor their growth. And I'll get into that in a minute. But first, we need to actually get our plasmid into cells. And how we do this is with transformation. So transformation is just a fancy word for like getting genetic info into bacterial cells. And so typically, we often do this with heat shock with competent cells. So we have these cells and they're really weakened. and we have this they have this calcium that is going to help neutralize the negatively charged phospholipids and the negatively charged DNA. They help them get close together. When you heat shock them, then you are going to kind of open pores in there and wider, and then your plasma is going to get in. And then you do a little outgrowth. So you add food without antibiotics in them, and then you warm it up in a little shaker to 37 degrees. So at this point is like the outgrowth. So you're kind of letting them recover. And then you're going to stick them in media with antibiotics so that you select, so you only let the ones that you want grow. So this is the basic transformation process. And we'll get to that so we ultimately want to stick them into our cells, the DE3. So with the T7 polymerase. But first we actually need to, if we're starting just like from the cloning phase, so where we just put this into our pet vector, we want to make sure that we actually did this right. So remember in molecular cloning, we're taking the instructions and putting it into a vector. And so like say our pet vector. So, but first we want to make sure that we did everything right, and we need to get lots of copies of this because we're going to want to get, later, we're going to want to have a stock of it, and we're also going to want to get it into our expression cells. So we need to have more of it. So if you imagine that you have you make one copy, but one copy is not going to be enough. So what you're going to do is first you put it into cloning cells, and these cells don't have the T7 polymerase. So they're not going to be able to make our protein, but that's okay because they're going to devote their energy to making lots of plasmid copies. So we can actually have some of these strains that we use, they have a high copy number, or they allow for a high copy number of your plasmid. So it depends on the plasmid and depends on the cell, but you can then they'll allow the cell to build up large levels of your plasmid. So you get lots and lots and lots of this. And then you can then plate them, and you can then, when you plate the bacteria, basically each of the colonies is going to be like a single bacteria that dropped down, made a home, and started multiplying on top of itself. So you get this gloopy colony where they're all genetically identical. You can take one of the colonies and stick it in some media, so some bacteria growth food, and a little tube of liquid. Let them grow overnight, make some more of themselves. Then you break them open, you lyse them, and you then check and make sure that the sequence is all right so that you did all the cloning well. So you want to do this in those cloning cells. So something like a DH5 alpha, something that doesn't have the DE3, something that doesn't have the T7 RNA polymerase, both so that you can get a higher copy number. You can have them not be worried about making protein, and you can have it so that even with the inducible expression system, those promoters can be kind of leaky. So you can also have protein made when you don't want to have protein made. And in addition to not wanting to just start things early, sometimes your protein might actually be harmful to the bacteria. It might be toxic or whatever. And so you don't want to be making it too soon. But now it's time that you can stick it into the cells that we want to stick it. So we can stick it into the expression cells, the cells that do have this T7 RNA polymerase. So these cells like the BL21DE3. And then later when we add the IPTG, we'll be able to de-repress it. So you clone it, you put it into your cloning cells, you make sure that you allow them to make lots and lots of copies of the plasmid. And then you sequence it, you make sure, maybe you do an analytical digest, or you do a colony PCR to get an idea of whether it worked, whether the gene's in there. And then you send it to sequencing to make sure it's okay. And so at this point, you have a lot of that pure plasmid. So once we have the plasmid and we know it's okay, now what we're going to do is we're going to want to transform it into those expression cells. And then what we're going to plate them. And so we're going to plate them on one of these Petri dishes. And so these are filled with this LB agar typically with the antibiotic, because we need the antibiotic in order to make sure to select for the ones that have the plasmid. So LB, lysogeny broth, it's basically just this basic form of bacterial food. It has some nutrients the bacteria need, and then the agar is a sugar that's going to let it form this gel, so that this gel filled with bacteria food that the bacteria can grow on top of, and they can make these colonies. Now what we want to do though is we're not gonna, like you saw those big flasks in there, we're not expressing our protein in these little colonies. We need a lot of cells and to make a lot of protein. So what we're going to do is we're going to take one of those colonies and we start an overnight growth. So we pick a colony off of the plate, and then we just take we do this with just like a micropipette tip. We take the tip and we just drop it into like 5 mils of growth media in one of these culture flasks. And then we're going to let it grow up overnight. And so this is like a starter culture. We can also just do it if we're doing a larger scale expression. We can do it in like a 25 or 50 mil volume in a flask. You can also go directly from, if you don't want to do this plating step, or you don't have time or whatever, you can take a transformation and you can just take some of the transformation mixture and dump it into there instead of doing it from a colony. But the colony, at least it'll be guaranteed to be genetically identical. And you also have more, you can keep the plate and use the other colonies later. Sometimes you even try a few colonies just in case. So you have this little starter growth, and the reason why we need a starter growth is because we can't just go from that one little colony and just dump that into 8 flasks, right? So instead we make this starter growth, and this is going to allow the bacteria to kind of build up high numbers, and then we can split it up between different flasks and not just overwhelm, not just like have a sufficient quantity that they can actually build up and start doing stuff at a reasonable time scale. So typically, and so at all these steps, you need to make sure you're adding your antibiotics. And so now you kind of have to, once you're ready to scale up, so the next morning you come in, now you're going to want to do a bigger growth. So you want to stick it into a larger flask. And so typically these flasks, we have these big flasks and we have like a liter in each of them. You want to make sure that the flasks aren't too full because you want them to have plenty of room to aerate. And you have to decide what kind of growth media you want to stick it into. So in addition to just having the antibiotic, so you want to make sure you have your antibiotic that matches your plasmid. You also want to make sure that you have the right, you also have a choice of media. So media is just like bacterial food. So the classic is like the LB that we talked about, but there are also variations such as TB, which we tend to, which is richer, this terrific broth. Sometimes it's better for protein yield, and its pH buffers prevent cell death. Then there's also like 2XYT. I have more on these in different posts, so I'm not going to go into it too much, but there are different media that you can choose from. And this will come into play potentially for your yield, and then also when we're measuring the growth. So we take that and we stick some of our starter culture into the flask. So somewhere between like 1 and 5 mils usually. Kind of depends, but doesn't impact things too greatly. Then you're going to want to monitor it so that you're inducing it when it's ready. So remember, we want to grow enough of the cells so that we have enough cells to make our protein before we start killing them. And so because they're going to devote themselves to making our protein and not to growing more, we want to make sure we have plenty of them grown, but we don't want them to overgrow. So when they overgrow, they're going to start running out of resources. And if they're running out of resources, then they're not going to have resources to make our protein. So we need to find this like optimal, the optimal point. And so we do this, we track its growth by measuring the OD 600. So basically we can do this with the spectrophotometer, yeah, I can never keep the terms straight, but basically you have a little cuvette. So this little rectangular plastic or glass little tube type thing, but it's square and it has clear sides. And they stick it in this little thing and it's going to shine light through it. It's going to shine light at this wavelength of 600 nanometers. And how much light gets through is going to be affected by how many bacteria are there. So you can think of the bacteria kind of like clouding things up, so not as much light can get through. And so this allows us to measure the growth of the bacteria. But we need to take into account that the media itself is going to impede some of the light transmission. And so we need to make sure we take a blank. So basically, before we add the bacteria into it, we need to take a sample of the media, and we're going to stick it in there and blank the spectrometer. So this is basically saying, okay, you're telling it this is background. And so now we're only looking at things that are above this level, because we don't want to get fooled into thinking that bacteria are there when it's really just the signals coming from our media. So typically what you do is you take like a ml or so of the media and you put it in here. And so because other people are going to be using this machine too, what you're going to do, you can like stick a piece of parafilm over the top. So parafilm's like this super duper cool, kind of like cellophane that's thicker. I love parafilm so much. But anyway, so you can stick parafilm around the top of this, and that way it's not going to like evaporate or that sort of thing. And then you just stick it on the top of there. And then, so if other people use it, then you can come back, you can blank it again. But you need to make sure that you take this blank before you add the bacteria. And so now you're going to measure the OD 600 over time. So over time, you're going to have more and more bacteria grow, and this is going to make it the OD higher and higher. And then once it reaches about 0.6 to 0.8 for LB, or about 1.4 to 1.8 for TB, then you can induce it by adding the IPTG. Typically, some add it up to like usually between like 0.1 and 1 millimolar of IPTG. It depends on the protein. So bacteria, they can fool you because they grow exponentially. And so at first you might think like, oh my God, they're being so slow. This is never going to grow. Like I have a meeting this afternoon. I'm going to be having to induce this like right in the middle of the meeting. But then once you wait, it starts like picking up because you have this exponential growth. And then sometimes you get fooled because it's like you think it's going super slow. So you don't take a time point. You're like, oh, I've got plenty of time. Then it starts like ramping up. And once it starts ramping up, it goes really, really fast. So once it gets close to one of those like values, typically you want to kind of close-ish, you want to start taking samples more frequently. Then once it gets really close, what you want to do is like by the time you prepare your IPTG and stuff, it's probably overshot. So typically what I do is I'm going to start getting it ready for things when it gets close. So what do I mean by like getting ready? So they're different depending on your protein, depending on the protocol. So look and see if people have expressed this protein before. What did they do? I tend to overexpress my things overnight, and I lower the temperature. So originally we're growing them at like 37, which is like they're optimal for growth and stuff. If we grow them at 37 and they're making a ton of protein, they're going to kind of get like exhausted or worn out or whatever. The cells can start dying and stuff, and not good. So if you let it go for a really, really long time. If you only let it go for like 3 or 4 hours, then you can do it at 37. But if you want to do like a longer expression, you can lower the temperature. So typically to like 18 degrees Celsius, and then let it grow overnight. That's what I typically do. A benefit of doing it at a lower temperature is that the bacteria, because this promoter is so strong and stuff, they can start making the protein like really, really fast. And if they kind of like go too fast, the protein can sometimes have problems folding, or if you make too much, then it gets misfolded, it aggregates, you get these problems. So lowering the temperature can sometimes help with if you have things like solubility issues or folding issues, that sort of thing. So either like overnight at a lower temperature or for a few hours at a higher temperature. So when I'm doing it at a lower temperature, once it gets close to the right OD, I'll start lowering the temperature on the incubator because it takes a while to get down there. So if I'm getting really close, if I take a sample and it's kind of close, then I'll just like lower the incubator temperature and let it start cooling down. If it is really close, then I'll take the flasks out and I'll actually stick them on ice in the cold room while I wait for the incubator to cool down. Then once the incubator is ready, then I can add the IPTG and then let things go. Before you add the IPTG though, you want to take like a sample, so a pre-induction sample. And so the pre-induction sample is before you add the IPTG. And the reason why you want to do this is because you can do a test with an SDS page to see if your protein was actually expressed. So to know if your protein was expressed, you want to see like, okay, well, what was there before I induced it, and after I induce it, I should see the protein. And how we're going to see this was with SDS page, much more on SDS page in other posts. But the basic idea is we separate proteins by size, and then we can stain in this gel. Then we can stain the gel for proteins, and we'll see bands representing the various proteins. So if you take a pre-induction sample and then you take a post-induction sample, so I'm not talking about like right after you do this, but what you're harvesting, when you're collecting the cells, if you take a sample, so typically I just take like a mill and I spin it down. So I centrifuge it to pellet all the cells, then I resuspend that and prepare a protein sample from there. Sometimes I do like a couple different concentrations and volumes and stuff, because it's really easy when you have this to like overload your gel because there's so many proteins in the sample. More on like the details and stuff in the posts. But so you take a pre-induction sample and you shouldn't see a band for your protein. Then you take a post-induction sample and you should hopefully see a band for your protein. If you don't see a band, it might not have expressed, or it might have expressed, but not super duper strongly. And so all of it may not be lost, and more on like troubleshooting and that sort of thing in other posts. And you can actually do like a Western blot to check if your protein's there, or the ultimate test is like purifying and see if you get anything. But because you have this over-induction, this overexpression, you can see the band. And so now it's time to harvest. Yeah, I mean, like you don't have to do, you're not doing the gel like before, like don't worry about the gel now. Take your induction, take a sample out, and then start with your harvesting, and then you can go from there. But with the harvesting, basically the idea is to collect the cells. So we want to remove all the media. So we pour the flasks into bottles, and then we centrifuge them to pellet the cells, and we remove the bacteria, the media, so the bacteria food. And so now we have a pellet with our cells with that protein. And then we're going to resuspend that in a smaller amount of clean liquid. This liquid, we have like protease inhibitors, so that once we break it open, we lyse it to purify it. It's not going to get all chewed up. And then, so we have it in this clean buffer. So it's like pH stabilized salt water. And then we can transfer it to a smaller tube and flash freeze it. And then once we're ready, so we have this tube of this pellet in our freezer. So we've got tons and tons of tubes of them. Most of them are, at least a lot of them are from insect cells, but same idea is from now on, it's like basically the same, except with bacteria cells, it's harder to like lyse them to break them open. So we basically typically do like a couple freeze thaws, tons of sonication. There's various different methods, but it's harder basically to break open bacterial cells is my point. But you break the cells open, and now you're gonna spin them even faster, and to pellet out the soluble stuff, which assuming that you're studying just like a soluble protein, so like one of them that just hangs out in the cytosol as opposed to like a membrane protein, then your protein is going to be in that liquid part. And then you can take that liquid part, so the supernatant, and you can then purify out your protein using various protein chromatography methods that I have much more about in other posts. So bacteria doesn't work great for everything. And so you can have problems with expression, especially if your protein is larger, more complicated, needs modifications, needs help folding. And so for this, we can turn to more like human-like cells if you're expressing like a human protein or whatever. Basically something that's eukaryotic. So something that has like that nucleus, it has more of the stuff that our cells have. And so one common way is with like insect cell expression using this like baculovirus expression vector system, or BEVs. And I have much more about that in yesterday's post. And I also have, I know that this insect cell expression is a lot more expensive. It's a lot more time consuming. Not all the labs have the capability for doing it. Bacterial expression, one of the things that's really great about it is it's good for I mean, it doesn't take a lot of fancy dancy equipment to do it. Just, I mean, like as long as you have like a shaker incubator and stuff. You don't need all the fancy stuff that you need for like insect cell or mammalian cell expression. So it's kind of easier access for labs around the world. Labs at smaller schools, you can do it in laboratory, like in undergrad classes, that sort of thing is really great. But sometimes it doesn't work well. That does not mean like all hope is lost. And so I have a post on things that you can do to kind of troubleshoot your bacterial protein expression. Things like lowering the temperature, as we talked about, lowering the IPTG concentration, checking whether it's pelleting out, whether it's not getting expressed at all, whether it's in the soluble portion, so much more in the like membrane portion, much more on that in that other post that I will link to. And happy expressing, and hope that your protein purifies well without having to resort to a lot of troubleshooting.